Assalamualaikum, good day everyone. So today we will begin with the last chapter on eukaryote and to wrap up on the whole course, which is on deregulation and cancer. So basically, well, if some of you, okay, or all of you are not aware, cancer is actually a multi-step process. So it doesn't occur just like that, but it occurs in a multi-step procedures. So we know that cancer are diseases that share a common overall phenotype. The phenotypes is an uncontrollable cell growth and proliferation, which means that when it grows a lot, so it invade and sometimes could basically cause neighboring cells to be cancer as well or cancerous as well. So cancer is a result of altered gene expression, and since it's a multi-step process, cells acquire a series of mutations that of course eventually lead to unrestrained cell growth, which means that uncontrollable, and division apart from unrestrained cell growth and division, inhibition of cell differentiation, and evasion of cell death. So certain cells after a certain time, they go through cell death apoptosis. So it doesn't occur if it's sustained, so it could sometimes lead to cancer as well. So as cancerous tumors grow in size, they would stimulate a process called angiogenesis, not to be confused with angiotensin, during which they acquire a new source of blood vessels that provides them with oxygen and nutrients. So eventually, the tumor cells invade the surrounding tissue and spread to other parts of the body in a process called metastasis. Now, cancer usually starts when a single cell acquires a series of mutations. So it can be either one after another, but eventually collectively it would change a once normal cell into a cancerous cell that would divide uncontrollably and of course may eventually spread throughout the body, again like I was saying earlier on. So gene mutations associated with cancer can be passed on through the germline, inherited from one's parent, such as breast cancer and prostate cancer, or sometimes they could be acquired through somatic mutations. So, progression to cancer can occur more rapidly if an individual inherits a germline mutation in a cancer associated gene. So, that's why, again, when you see that when someone has a breast cancer, prostate cancer, and if the children also has it, the children usually have it much worse as compared to the parent or to the predecessor. So what happens is that this person's cells will be one step closer to cancer from the very beginning. And of course, if they don't take care of themselves, are not aware of it, so it could progress quite fast. So among the currently known cancer genes, 70 are associated with germline mutations and 342 are associated with somatic mutations. So can you imagine that there are a lot of cancer, and one cancer basically there are more than one genes involved. So basically in this diagram, again, this is how uh, this is a depiction of this 5-hit scenario for colorectal cancer. So this is how mutations could occur from normal colon cells, okay, whereby in the beginning it has two APC mutations, whereby it later on can develop a one RAS mutation to basically produce an adenomatous polyp. So it then progresses to diplastic polyp, with two TP53 mutations. Okay, so later on, as it progresses, it divides uncontrollably. Okay, so we got colon carcinoma, and other events could also occur, such as chromosomal aberrations, which means that uncontrolled cell division. So this would later lead to metastatic carcinoma, which means that cells that have progressed into cancer and probably would have been erupted or spread to other parts or nearby cells. So, a gene that is not normally expressed in a cell usually can be switched on and expressed at high levels, and this could result in gene mutation or changes in gene regulation, such as epigenetic variation, transcription, post-transcription, translation, or post-translation. That's why we have learned everything about transcription and a bit of the translation in this course. Okay, and this is basically how gene mutation could occur and sometimes lead to cancer. So changes in epigenetic regulation, transcription, RNA stability, which we have covered, protein translation, and post-translation control can be detected in cancer. So while these changes do not occur simultaneously in one cancer, the changes at each of these levels could be detected. This is when you observe cancer at different sites in different individuals and depending on the type of cancers. So, changes in histone acetylation, which basically an epigenetic modification that leads to gene silencing or activation of transcription factors by phosphorylation or increased RNA stability or increased translational control, and not to mention protein modification, can all be detected at some point in various cancer cells. So, how do you regulate gene expression in cancer? So you need to know different terms, okay, that is associated with cancer. We have carcinogenesis. Carcinogenesis is a term used for normal cells that has been transformed into cancer cells. So the process, okay, is known as oncogenesis or tumorigenesis. There are two processes, okay, whereby carcinogenesis would occur via oncogenesis and tumorigenesis, which we will go through one by one again in a while. So whether it goes through oncogenesis or tumorigenesis, it depends on both the activation of oncogenes and deactivation of tumor suppressor genes. So now you know oncogenes, okay, is actually caused by, oncogenesis is caused by oncogenes and tumorigenesis is caused by tumor suppressor genes. There is a defect in them causing them, okay, to progress to carcinogenesis. So apart from that, okay, we have a proto-oncogenes, okay, which is a non-cancerous normal version of an oncogene, which could be a transcription factor like the CFAS, CJUN, and CMYC, or a gene whose product is involved in signal transduction, which could lead to an altered gene expression. So when mutations cause the regulation of this process to be disturbed or altered, cell proliferation can be enhanced, leading to tumorigenesis, which means that later on, okay, it couldn't be controlled. So at least two separate mutations are necessary to develop cancer, as we know, oncogenesis and tumorigenesis. So mutation in a proto-oncogene would not necessarily lead to cancer, as normally functioning tumor suppressor genes would counteract the effect of the oncogene. So that is the function of tumor suppressor genes, as the name says, to suppress the effect of oncogenes. So mutation in tumor suppressor gene, on the other hand, could lead to uncontrolled cell growth and possibly cancer. So if your tumor suppressor gene couldn't do what it should do to suppress the tumor, so it now could lead to tumorigenesis or it could progress to cancer. So gene expression, signal transduction, the cell cycle, and oncogene seems to have an interconnected relationship with each other inside the cells. And well, by right, they should, and they do have. So numerous oncogenes are known to be involved in gene expression. So what we term as oncogenes, okay, we will come to that. So signal transduction pathways are filled with proteins involved in ultimately altering gene expression. So anytime one of the involved proteins is altered, so such as when the gene is mutated, gene expression will also be altered. So of course, okay, when gene is mutated, okay, gene expression is also going to be altered depending on the mutation that occur. Now, this is a kind of like a diagram on how cancer develops. So it shows a series of mutations in tumor suppressor genes and proto-oncogenes, okay, leading to cancer. So we know that there are two causes, okay, that could lead to cancer: tumor suppressor gene or oncogenes. So usually, if there is a defect, okay, on proto-oncogenes, okay, it could lead to oncogenesis. So a mutation, okay, that occurs to tumor suppressor genes, okay, would inactivate them, which means that they now couldn't function to suppress tumor. So what happens, okay, is that cells would proliferate, okay, whereby when they function properly, okay, by right, they should control the cell proliferation or inhibit proliferation of tumor. But now, since there are mutations, so tumor suppressor is inactivated. So cells now proliferate. Mutation, okay, would inactivate DNA repair gene, which means that no DNA repair mechanism could repair the condition. And somehow, okay, it could also either be added with mutation of proto-oncogenes, okay, which of course we know create oncogenes, which cause a defect, okay, and adding, okay, to the cell proliferation or uncontrolled cell proliferation. So if an uncontrolled cell proliferation occurs, okay, it could cause metastasis. Okay, so mutation would inactivate several more tumor suppressor genes. Now, well, the condition is uncontrollable, so you got metastasis, which means that, okay, it's now progressing to cancer. So the genes that drive cancer are tumor suppressor genes and proto-oncogenes. So mutations in these two genotypes could lead to cancer. So proto-oncogenes, okay, so remember this, okay, a few while back, okay, act as gas pedals to accelerate cell growth and division. Tumor suppressor genes act as brakes to inhibit cell growth and division. So mutations in either one of them, okay, for example, that inhibit activity of overactive proto-oncogenes, okay, hit the gas pedal or tumor suppressor gene, cells lose their brakes, can drive cells to cancer. So in the end, the result is the same: uncontrolled acceleration toward a cancerous state. So tumor suppressor genes, okay, is first discovered by Alfred Knudsen in 1971, and mutations that inactivate the tumor suppressor genes are called loss of function mutations, and they could be caused by these two factors. Deletion of the tumor suppressor genes, okay, usually caused by point mutation, whereby it could cause a functionally inactive tumor suppressor gene encoded protein, which means, of course, the result would be that the cell would proliferate without control. And second, mutation that disrupts the promoter region of the tumor suppressor gene. This could lead to a cause or a loss or reduction in its expression, which is not good either way. So, tumor suppressor gene, again, loss of function, okay, usually are recessive, and both copies of cell's tumor suppressor gene must be mutated in order for cancer to occur. And that is basically what contributes to cancer. Okay, usually both of the alleles are mutated, okay, progressing to cancer. So what controls a growing tumor? There are two main types of gene, which we know, proto-oncogenes and tumor suppressor genes. So we know that tumor suppressor genes are supposed to suppress, okay, a growing tumor. But if a mutation occurs, it inactivates the gene, which means that it won't slow or control the growing tumor, okay, it would accelerate it. What happened with proto-oncogenes? So proto-oncogenes, okay, is also a form, it sometimes could be in a form of transcription factor. So when transcription does not occur properly, detrimental effects can result, and the result is cancer. So what are oncogenes? So they are the products of proto-oncogenes, which are required for normal growth, repair, and homeostasis. But when these genes are mutated, they turn into oncogenes. Okay? So the good genes are called proto-oncogenes, okay, but once they are mutated, they are termed as oncogenes, okay, and play a role in the development of cancer via oncogenesis. So proto-oncogenes may be either growth factor, transcription factors, or other proteins involved in regulation, transcription or translation, but usually doing transcription. So a very common oncogene, okay, called RAS, which is a regulatory GTPase, okay, that switches the signal transaction chain on and off, is uh, usually an important protein, especially because they are product of oncogenes which are found in 20% to 30% of human tumors. So chances are a lot of human tumors, okay, is actually caused, okay, by this RAS and RAS-related proteins. So the transcription, another transcription factor MYC, okay, is also an oncogene that is often seen mutation in Burkitt's lymphoma, which is a rare type of lymphoma, a cancer of the lymphocytes, okay, or your white blood cell. Now let's have a look, okay, at the RAS protein. So RAS G protein, okay, usually can either be inactive or inactivated form to regulate the GTP hydrolase. So when a growth factor binds with receptor on the outside of the cells, a signal would be relayed to RAS protein, and RAS is activated when GTP is bound to it. So an active RAS then passes the signal to a series of protein kinases. So remember your two component, kind of like your two components. So regulatory proteins that eventually activate transcription factor to alter gene expression, and of course, again, it will produce proteins that simulate, okay, the cell cycle. And one important recipient of RAS signaling is the mitogen activated protein kinases, MAPK, which is basically in abundance, implicit. So once activated, MAPK transmits signals downstream to other protein kinases and gene regulatory proteins in the signal transaction pathway. Many of these genes and proteins, of course, again, is involved in signal transaction pathways, and they are interconnected to RAS. Okay? Like I said, okay, RAS is actually quite an important protein for transcription. So any mutation that makes RAS more active or otherwise interrupt the normal signal transduction pathways may result in excessive cell division and eventually lead to cancer. So more or less, okay, this is what happens, okay, when RAS, okay, or mutation or disruption, okay, in the RAS G protein. So this is the condition, okay, that basically I've shown you earlier on, okay. So this is a depiction, okay, of the diagram. So basically, if it is involved, okay, as a growth factor, so usually, okay, your RAS protein, okay, basically should relay or should be passed on, okay, to other protein kinases, okay, and in the nucleus, okay, with other transcription factor, it would activate a gene and of course, okay, eventually a protein. So the protein, okay, would stimulate the cell cycle. But, okay, if a mutation occurs, okay, so RAS protein would be active with or without a growth factor. So what happens in the nucleus is that okay, there would be an overexpression of protein, and overexpression is usually not a good thing because well, in life there have to be a balance. Okay, nothing should be too much or too little. So this is a very complex pathway of the rest. So don't worry, you guys don't need to remember this. Okay, I'm just putting this up because the rest is involved. Okay, in a lot of gene regulation. Okay, as you can see over here. Okay, gene regulation in the nucleus. Okay, and especially okay, around the cytoplasm. Okay, in a lot of organisms. Especially, of course, we know in eukaryotes. So apart from that, it is also involved in apoptosis or cell death. Okay, apart from cell proliferation. So this is a part of the signal transduction pathway. We know that RAS okay, activates a number of pathways, but and especially important, MAPK. So MAPK usually transmits signals downstream to other protein kinases. So your RAS okay, will usually act okay, up here. So note that many of these pathways are initiated when a single signal binds to its receptor outside the cell. So it would end up okay with an altered gene regulation and cell proliferation if the RAS protein is being inactivated or mutated. So let's move on further with the oncogenes. So a few cancer syndromes are caused by inherited mutations of the proto-oncogenes, which could later cause the oncogene to be turned on or activated. But again, as with the case of oncogenes, again, most cancer-causing mutations involving oncogenes are usually acquired, not inherited, which is actually well, a bit of a good news, okay, because they generally are activated, okay, oncogenes by either chromosomal arrangement, which means that changes in chromosomes that put one gene next to another, which allows one gene to activate the other. How it does that is actually a very complex mechanism, or gene duplication, okay, having extra copies of a gene. Okay, like I said again, extra anything is not good as well, which can lead to it making too much of a certain protein, which would be detrimental and could lead to cancer. So the other component okay, to cause cancer is the tumor suppressor genes, as we know. So its function, of course, is to suppress tumorigenesis or control the tumor formation. But when the process is disturbed, usually by mutation, tumor suppression may not be inhibited as normal, which means that well, your tumor could now probably grow as normal or probably double up. So tumor suppressor genes are normal genes that slow down cell division. Remember the break. So they repair DNA mistakes again and tell cells when to die, a process known as apoptosis or programmed cell death, PCD. Kind of like a lot of our cells okay, in the body. So when tumor suppressor genes don't work properly, cells could grow out of control, which could lead to cancer, which means that there is nothing okay, to slow the growth. Okay, it's now controlled okay, happily or without anyone telling them to stop. Okay, so it can grow. So the genes usually would normally keep the cell from dividing too quickly, just as a brake keeps a car from going too fast. But when something goes wrong with the genes, such as mutation, cell division can get out of control. So the one difference okay, or two difference okay, as you can see over here between oncogenes and tumor suppressor genes is that oncogenes result from the activation or turning on of proto-oncogenes, but tumor suppressor gene is the opposite. Okay? It causes cancer when they are inactivated, turned off. Either way, the result would be the same cancer. So inherited abnormalities of tumor suppressor genes usually were found okay, in some cancer family cancer syndromes or inherited. So they cause certain types of cancer to run in families. But most tumor suppressor genes mutation are usually acquired as well, again not inherited, unless for certain breast cancer and prostate cancer. So an example of tumor suppressor gene, the most famous is actually P53, which encodes a 53 kilodalton protein. So P53 gene is activated by the DNA damage caused by ultraviolet light, UV light, and any damaged DNA which may be harmful to the cell. So to make sure that the damaged DNA is not replicated, because then everything would go haywire in your body. So the cell cycle must be temporarily stopped so that the DNA can be repaired. So the P53 tumor suppressor gene encodes a transcription factor, which regulates the synthesis of cell cycle inhibiting proteins. And it also often activates a gene named P21, okay, whereby okay, it produces a protein okay, that temporarily stops the cell cycle. So if the DNA cannot be repaired, p53 would activate other genes that could lead to cell death or apoptosis to prevent the cell from passing on damaged DNA, because it's not good for the cells, for the system. So if the basically p53 tumor suppressor gene is defective, for example via mutation, DNA damage in the cells may be accumulated and the cell may survive to be replicated. Okay, and of course, okay, would be distributed, okay, not only from the origin, but could be passed on to nearby sites. So the damaged DNA would then be passed on to other cells, like I was saying, through cell division, and would basically develop into cancer. So this is basically kind of like the damage, okay, from DNA that basically YP53 as a tumor suppressor gene, which is very crucial to make sure that the damage doesn't get out of control or doesn't get replicated or should basically be removed from the cell. So if it is not removed, okay, what would happen is that, well, you can get an overgrowth, okay, or survival of the damaged DNA, which is, well, the result would be detrimental to the whole cell. Alright, so I'll leave you to that for the first video. So move on to the second video for cancer and transcriptional control.